This is Team 1's presentation on the article of How Dynamic Remodeling of Membrane Composition Drives Cell Cycle Through Primary Cilia Excision by authors Su Cheng Fua et al. What is cilia? You may remember the hair-like structures inside your lungs to help move debris and microbes, or as appendages on the ends of cells to help them be motile. These are cilia. These structures are evolutionarily conserved in eukaryotes. They are made up of microtubules. They evolved from single-cell organisms 1.5 billion years ago. There are many categories of cilia, but this paper focuses on primary cilia. Primary cilia help cells receive signaling molecules, for instance, to help them with cell division. They also help orient or coordinate the cell's planar cell polarity. They're generally used to help gather information about the cell's environment. The significance of this research will help researchers understand the normal processes of cell proliferation or cancers that arise from mutations within these cellular and molecular pathways of cell division. Also, it can help us understand and treat serious genetic developmental diseases such as Joubert syndrome and Morm disease, which are linked to the cilial enzyme INPP5E. Introduction. Overall, this paper is trying to understand the molecular pathways and mechanisms that manage the primary cilium life cycle during disassembly. The cilia life cycle is coupled with the cell cycle. Cilia structures are formed in non-dividing cells and disassembled when entering the cell cycle. Primary cilia disassemble by gradual resorption of ciliary material back into the cell. It is unknown how cilium is sequestered from cell surface functions such as vesicle transport and how it maintains its stability through these membrane processes. The enzyme PI45P2 phosphatidylinositol 4,5-bisphosphate is involved in almost all membrane processes. This enzyme brings phosphatidylinositol 4,5-bisphosphate binding actin regulators to promote actin polymerization inside the cell cortex that will stimulate cell surface membrane processes. As you can imagine, the plasma membrane is adjacent to the cilia membrane. To molecularly distinguish these areas in the cell, phosphatidyllinositol 4-phosphate, PI4P, is highly present in the ciliary membrane, while phosphatidyllinositol 4,5-bisphosphate is present in the surrounding plasma membrane. The enzyme INPP5E, phosphoinositide 5-phosphatase, compartmentalizes phosphatidyllinositol 4-phosphate and phosphatidyllinositol 4,5-bisphosphate in their appropriate membranes. Cilia-enriched INPP5E loss of function has shown in previous studies to accelerate cilia disassembly and replenishing phosphatidyllinositol 4,5-bisphosphate, associating phosphoinositides to primary cilia stability. Aurora A kinase induces cilia disassembly and absorption via destabilizing acetylation of microtubules by histone deacetylase 6, HDAC6. Previous research in the field has shown there are releases of vesicles from distal cilia in INPP5E mutant mice. This leads to a connection between phosphoinositides and extracellular vesicle formation. With all of this background research, the researchers now have a good place to find out the molecular mechanisms and molecular pathways involved with cilia disassembly and the cell life cycle. The first experiment the researchers performed was to find conditions that promoted primary cilial vesicle release. NIH3T3 cells, HT-ERT-RPE1 cells, and MICD3 cells were put into a quiescent state by culturing them in a 0 to 0.1% fetal bovine serum for 24 to 48 hours, inducing ciliogenesis. The researchers used 5-HT6-mCherry GFP and ARL13B GFP markers on 3T3 cells and RPE1 cells respectively to tag the cells and visualize potential ciliary membrane vesicle release. After a few hours, the tips of cilia can be seen thinning and excised in figures A and B. in cell cultures 3T3 and RPE1. In the 3T3 cells, you can see that gamma tube GFP marks the centrosome, which is the organizing center for microtubule assembly. Knowing that loss of function in INPP5E has shown to accelerate cilia disassembly in mice, researchers compared a loss of function mutant and non-mutant INPP5E mouse fibroblasts. Researchers found that in quiescence, cilia was shorter in the loss of function, had a lower concentration of cilia, and had a 68.3% plus or minus 6.3% occurrence of vesicle release, as compared to a 23.8% plus or minus 1.5% vesicle release in a non-mutant INPP5E in a 3-hour period. As you can see in figure 1C and D, red arrows are cilia, yellow arrows are excised tips. As you can see in figures C and D, there's also 10% FBS added. This is a growth-stimulated culture. Inducing growth stimulation in a non-mutant INPP5E caused a higher vesicle release of 65.5 plus or minus 5.3%, yet the loss of function mutant did not have any higher vesicle induction. You can see the little vesicles flying off in the yellow arrows, but what happens to the vesicles with excised cilia tips? They can be found associating with the cell surface by comparing the proportion of ARL13B marked particles in acetylated alpha-tubulin. Remember, acetylation of microtubules by HDAC6 is how microtubules are destabilized. We can see in figure E, it shows that the cell surface is elucidated in GFP, with the particles of ARL13B labeled as cilia bound to the surface, as you can see in a 2D and 3D reconstruction. In figure F, you see acetylated tubulin in red and excised cilia in green from ARL13B label and DAPI stained nuclei. You can see in the first non-mutant picture, cilia are long and near the nuclei and undetached. In the loss of function mutant picture to the right, you can see the dots of excised cilia with acetylated alpha-tubulin going to the surface. G gives us a significant difference in percentage of cells with acetylated alpha-tubulin and GFP-labeled cilia in our mutant and non-mutant mice fibroblasts. Next, there was 24-hour cultured media with either 0 to 0.1% FBS, which was quiescent, or they had growth-stimulated cultured media, which was 10% FBS, and these cells were then analyzed. MIMCD3 had higher levels of ARL13B and growth-stimulated media, yet alpha-tubulin and GADPH levels are unaffected by growth stimulation. H shows the Western blot of the proteins, and I shows the measured intensity in graph form. How you interpret the Western blot here with the proteins is that they're spinning down a cell lysate, which is going to remove the soluble proteins because they're going to be putting the pellet into the Western blot gel. And so as you can see with the cell lysate, alpha-tubulin is present, the ARL13B is present, and the GAPDH is present because this is all one big soup. And then when they spin down the cell lysate and get the pellet, soluble proteins are not pelleted. So alpha-tubulin in our control that we know is a soluble protein, GAPDH, they're removed from the pellet. But we still have ARL13B present in a very intense band. And so that shows us that this protein is associated with this cytoskeleton because the pellet proteins, the proteins present in the pellet are associated with the cytoskeleton or the cell surface. So this confirms that ARL13B, the part of the cilia that is excised from the tip, is associating with the cell surface. And this is also seen graphically in I. With all that information, the researchers can conclude in the first part of the paper that ciliary vesicle release is promoted by INPP5E loss of function and growth stimulation. Next, the researchers explore the relationship between INPP5E and growth stimulation. Mouse embryonic fibroblasts were in 0% fetal bovine serum or 10% fetal bovine serum for 4 hours to measure the intensities of acetylated tubulin and INPP5E in figure 2A and B. There was significantly more INPP5E in 0% fetal bovine serum, which is the quiescent medium. Researchers were concerned that lower levels of INPP5E may cause a lower depletion of PI45P2. Looking at the figure, 5-HT6 is a protein specific to primary cilia used to conjugate to a fluorescent protein for cilia visualization. PHPLC delta binds to PI45P2 and is also conjugated with a fluorescent protein for PI45P2 visualization. In the last row, it is an overlap of both images. When PI45P2 was confined within the proximal half of the cilia in quiescence, INPP5E non-mutant mice embryo fibroblasts and 10% of fetal bovine serum led to PI45P2 accumulating in the distal half of INPP5E non-mutated cilia, like what happens in cilia excision. Looking at the bottom of figure C, you can see that the PI45P2 is congregating at the bottom of the cilia. The cilia is in red, and you can see from the arrows that the PI45P2 congregating at the bottom of the cilia at the distal location. Relative accumulation of PHPLC delta, which binds to PI45P2. The relative accumulation on the distal versus the proximal side is shown over time on a graph in D. Now that last experiment, when looking at figures C and D on figure 2, those were done in 0% to 0.1% fetal bovine serum. So that experiment was done in a quiescent state. They redo the experiment of tracking PI45P2 via fluorescent tags in the cilium to track its location, whether it's congregating at the distal or proximal location of the cilium. So they redo the experiment using 10% fetal bovine serum in a growth stimulated environment. And so you can see, looking at figures E and F with the arrows of the overlap pictures, that the fluorescently tagged label PI45P2 is also congregating at the distal location in the cilium based on the arrows. You can also see in figure F there's a more drastic representation of how the relative accumulation of the radiolabeled fluorescently tagged label is congregating at the proximal location and going away from the distal over time. The red diamond indicates when there is excision of the cilia decapitation head. In graph G, we are measuring location of the fluorescently tagged PI45P2, whether it is in the proximal or distal location of the cilia. We are also measuring if there was cilial decapitation or not. The percentage of decapitation occurrence, whether distal or proximal, is shown for both quiescent and growth-stimulated media over a 2-hour period. As you can see, the majority of decapitation is happening in a growth-stimulated media in a distal part of the cilia, as you can see from the red bar. Looking at H, it's shown in another figure, the relative site of excision by relative PI45P2 accumulation. And as you can see, this is also mostly in the distal areas of the cell. With all this information that they showed in the figures, this brings the researchers to conclude that here growth stimulation regulates ciliary INPP5E and PI45P2 distribution throughout the cilia. So when there's growth stimulation, IPP5E will go away, and PI45P2 will be distributed into the distal part of the cilia, which is correlating with cilial decapitation.
Alright everyone, it's my turn now. Looking at what we know about PI45P2, also by the way I'm just going to call this PI4 from now on, so remember when I say PI4 I mean PI42P2 or
Phosphatidylinoacetyl 4,5 bisphosphate.
Because I'm tired of saying all of these big words. So we know that PI4 could affect ciliary localization of actin regulators. So the researchers looked at how F-actin was involved in all this, especially with primary cilia of INPP5E positive and negative MEFs. So they directly measured the amount of PI4 by, quote, targeting constitutively active PI45 kinase LG-PIPK to cilia 5-HT6-PF-PIPK. Basically using PIPK to amplify the amount of PI4 in the cilia. It's not clear how they did this, but regardless they found a higher amount of F-actin present. Time for a diagram. Here's a diagram. As you can see by the blue stars, because originally I planned to animate all this, but then I got lazy, so I'm just adding in random shapes now to kind of show where all my stuff is. Here's where the F-actin is present in the primary cilia. F-actin is shown by the white triangles inside the blue stars. And here with the pink rectangle, there's no F-actin as there's no white triangles inside them. Note that there's no F-actin in the samples with PIPK-KD, which had the researchers conclude that PI4 exerted direct control over F-actin assembly. The next step was to see how F-actin played with cilia decapitation. The researchers made some more INPP5E positive and negative MEFs and left them for a few hours, seemingly doing the same experiment again.
Here we go again.
No surprise, the researchers saw F-actin was present as shown by the blue stars. Then the researchers wanted to see how important F-actin was to all this. So they made an experiment to limit F-actin with some key players.
Litrunculin A,
which restricted growth-stimulated ciliary vesicle release in INPP5E positive and negative MEFs. Thymosin B4, which sequesters G-actin, which prevents polymerization into F-actin. And blebistatin, which inhibits myosin 2 to suppress growth-stimulated ciliary vesicle release in INPP5E positive and negative MEFs. I'm going to compress all their info together. So basically, as you can see with these diagrams, the samples without all the stuff that messes with F-actin are quite thick. And the vesicles present in dark blue are the ones with it and are quite small and have fewer vesicles. Looking further, even the amount of cells with YFP positive particles, basically vesicles, was reduced when they were put with stuff that messes with F-actin. All this stuff confirms in a very redundant way that F-actin affects primary cilia excision. The next step was to see what was in all these vesicles that were being released. So it was time for another experiment. The researchers, somehow it doesn't say exactly, dissected the vesicles after conditioning some of them for 24 hours with quiescent or 0.1 FPS, plus growth stimulated, i.e. 10% FPS, wild-type media. Remember from past slides that this stuff increases cilia decapitation, and the rest in cilia-deficient IFT88 knockout MLMCD3 media. They then performed various analyses like mass spectroscopy and global proteomic profiling. After doing this, the researchers found 1,376 proteins that were detected at least twice in conditioned media of at least one experiment condition. Of these, only 477 were proteins that were extracellularly released thanks to the growth stimulation. 71 of these were released due to functional cilia, which leaves 57 proteins as being both dependent on growth and cilia. Figure A sums this up. By the way, this figure was such a pain to decipher initially. It took us several days to understand what was even going on. Anyway, figure
B
5 shows us that what was in these vesicles that I spent the last several paragraphs stalling for time for. Mostly some large intraflagellar transport-related proteins, a subset of ciliary proteins, and hedgehog signaling effectors. The ones labeled with IFT-B were found to be way more abundant than IFT-A. You can see this in figure C and D, as there's way more green stuff than there is red stuff. I even drew arrows highlighting this. Going deeper, the researchers found that even in the protein level, there was way more IFT-B stuff like ARL13B, IFT88, and IFT81 that was detected, whereas IFTA stuff like IFT122 and IFT140 were barely even present. The researchers made figures E and F to try and summarize all this. So, what does this all mean? It basically means that when you do growth-stimulated ciliary vesicle release in primary cilia, it's IFT-B and its friends that get the boot rather than IFTA.
Moving forward with the notion that ciliary decapitation results in a substantial loss of proteins from the primary cilia and that this loss could compromise the integrity of the cilia, the researchers wanted to look at how decapitation affected ciliary reabsorption into the cell in INPP5E positive and INPP5E negative cells over the course of 3 hours in different strengths of growth media. And so here are the graphs of the relative cilia lengths observed over the course of the 3-hour window. They defined 1 to be the length of the cilia at time 0 and used that as a basis of comparison for the cilia length after 3 hours. The 2 bars on the left of each graph are ciliary lengths in cells that showed no decapitation in the 3-hour window. The 4 bars on the right of each graph show the relative cilia lengths when time equals 0, then prior to excision, then post-excision, and finally at the end of the 3-hour window. So what the researchers took from this was that in any condition that stimulated decapitation, be it by IMPP5E negative or by growth media, that significant cilia reabsorption occurred, which can be seen by the furthest right bar in each graph. It should be noted that in figure B, the upper right corner shows a 0 in the end of the 3-hour window. This is the effect of both growth media as well as from thymosin beta-4, which suppresses cilia decapitation by preventing G-actin from polymerizing to F-actin, and as such, only one of the cells was shown to have cilia excision. And this cell showed full cilia reabsorption before the end of the 3-hour window. Additionally, in the bottom right corner, we have INPP5E negative cells with no growth media and wild-type thymosin beta-4, which does show reabsorption, but decapitation was only observed in 3 of the 14 cells. Next, researchers looked at the role that decapitation played on the amount of ciliation in INPP5E positive and negative cells, grown in 10% fetal bovine serum, and under the effect of thymosin beta-4, which prevents actin from polymerizing. As can be seen in both INPP5E positive and negative experiments, all 3 groups had similar proportions of ciliated cells at time equals 0. But that over time, the 5-HT6 and mutant-type thymosin beta-4 expressing cells showed a decrease in overall ciliation. However, the wild-type thymosin beta-4 expressing cells was able to maintain its level of ciliation in the 20-hour window. This showed that inhibition of ciliary actin by the mutant-type thymosin beta-4 suppressed decapitation, and as such, saw less ciliary disassembly. Next, since growth stimulation would cause quiescent cells to enter into the cell cycle, researchers wanted to look at the timing of the cilia decapitation in relation to the cell cycle. To do this, they used a marker called Venus P27K, which identifies cells in G0 in green, as well as mCherry HCDT1, which identifies cells in both G0 and G1 phase in red. This would allow for distinction between cells in G0 phase, which
would cofluoresce green and red, which shows up actually more as a yellow, and G1, which would only show up as red, and in S1 phase, which shows up as no fluorescence. As well, they labeled the primary cilia with a cool electric blue color. So with this image, you can basically ignore everything except the row second from the bottom in the red box. So here what you're seeing is quiescent cells subjected to growth media for 10 hours. Moving left to right in box 1, we can see that the nucleus is yellow, indicating the cell is in G0 phase, and that the primary cilia in blue is intact. In box 2, we can see cilial vesicles as indicated by the white arrows showing that decapitation has occurred, while the nucleus is still showing to be in G0. In box 3, we can see the nucleus getting less yellow, more of a red-orange color, indicating entry into the G1 phase. And in box 4, we can see the entire nucleus is red, indicating the cell is now in G1. And in boxes 5 and 6, we can see that the cell has entered S phase. This is further illustrated by the graph on the right, which represents the relative amount of venous P27K as a function of time. The researchers decided that the point at which the signal from P27K was at 50% of its original would indicate the transition point between G0 and G1 phase. The red diamonds along the line indicate the times at which the cilia decapitation occurred, which, for 19 of the 20 cells that observed decapitation of the primary cilia, this occurred prior to the G0 G1 transition, which took on average 5.7 hours, showing that growth stimulation during quiescence induces cilia decapitation in G0 phase. The researchers next wanted to look at the effect of inhibiting cilia decapitation, and if this would have any impact on the cell's transition from G0 into G1 phase. To do this, they performed the same experiment as before, except the cells observed were expressing wild-type thymosin beta-4, which prevents decapitation by preventing actin from being able to polymerize. It is shown here that despite being grown on growth media, there are no cilia decapitation events, and only 70% of the observed cells exited the quiescent state. And of these cells, 57% were able to reach the midpoint, indicating entry into G0 G1 transition phase. This also took an average of 8.9 hours, whereas the previous experiment took only 5.7 hours. Ultimately, this showed that cilia decapitation is required for timely entry into the G1 phase of the cell cycle. Next, because the primary cilia is the central location where hedgehog signaling occurs, the researchers speculated that cilia decapitation might be promoted through mitogenic hedgehog signaling. So for those who aren't fully aware, here's a quick and very simplified summary of hedgehog signaling. So it's a signaling pathway that operates through the primary cilia and is activated when an external signal binds a protein called patch, which ultimately leads to the activation of a protein called smoothened. Activated smoothened results in these proteins called Gli proteins being able to enter the cell nucleus, where they act as transcriptional activators for particular genes. When hedgehog signaling is inactive and smoothened is deactivated, the Gli proteins are cleaved, and this cleaved version of Gli is able to enter the nucleus, where it's able to act as a repressor for particular genes. So what the researchers did was use a smoothened agonist called SAG, which causes smoothened to be constitutively active to see if this played a role in decapitation. This would lead to persistent activation of Gli proteins, which act as transcriptional activators for a green fluorescent protein. And increased levels of this green fluorescence would indicate activated hedgehog signaling. All the while, the researchers would look at cilia length while this was occurring. What they saw was that in all three cell types used here, there was initially a low basal level of GFP in the different cell types at the 8-hour mark. but by the 24-hour mark, the amount of GFP seen had dramatically increased, indicating a persistent activation of smoothened and the hedgehog signaling pathway. However, what was observed was that in the cells treated with SAG, the smoothened agonist, there was not significant decrease in cilia length, showing that the hedgehog signaling is not causing the decapitation. This was confirmed by treatment with vismodegib, a smoothened antagonist which inhibits the hedgehog signaling pathway and as such should show a decrease in the levels of green fluorescent protein. Vismodegib was able to prevent the green fluorescent protein fluorescence increase seen in the SAG-induced conditions. But as you can see, vismodegib was unable to decrease the fluorescence significantly in cells that were growth stimulated by fetal bovine serum. These results all indicated that cilia decapitation occurred independent of the hedgehog pathway. To further verify this, the researchers looked at mRNA levels of PITCH1 and GLI1 genes under growth stimulation by FBS, as well as by treatment with the smoothened agonist SAG. As can be seen here, FBS was able to only effect a slight change in the levels of PITCH1 transcription over the course of 8 hours, while GLI1 transcription showed relatively no change. However, under treatment of 200 nanomolar SAG, the levels of both PITCH1 and GLI1 gene transcripts showed a huge increase in transcription levels of both PITCH and GLI. So in summary, FBS, fetal bovine serum, causes decapitation, but does not activate smoothened, and SAG activates smoothened, but does not induce decapitation. Together, this indicated that cilia decapitation is not regulated by the hedgehog pathway.
To conclude, growth induction displaces ciliary INPP5E. It accumulates phosphatidylinositol 4,5-bisphosphate in the distal cilia, and this causes actin polymerization in primary cilia and excises cilia tips in a process called cilia decapitation. There's acute loss of IFTB through cilia decapitation. It precedes resorption. Cilia decapitation induces mitogenic signaling, and there's a connection between the cilia life cycle and the cell division cycle overall.